Good evening. My name is Howard Edelman. I'm your host for Israel and the Jewish World. Howard Cedar was awarded the prestigious Israeli Wolf Prize in Medicine. He has been touted as a future candidate for a Nobel Prize as a leading researcher in methylation. If DNA is methylated, it is open and sensitive. If unmethylated, it is closed and inaccessible. In epigenetics, he demonstrated how DNA operated as a regulatory mechanism. That is why our show is called Switching, because a key issue is when and how genes are turned on and off. Does biology provide insights into sociology? When do Jews get turned on or off their religion? How do Jewish youth get turned on to stand up in defense of Israel? I interview a former IDF officer, now a student at UBC, who has become active on campus. Stay tuned. This is another in our series on medical innovation in Israel and focused on the Institute for Medical Research, Israel, Canada. My guest is Howard Cedar. Welcome to the show, Howard. It's Howard or Chaim. Which do you prefer?
Chaim.
Okay, Chaim it is, because I'm a Chaim, you're a Chaim. Though in Canada I'm known as Howard. You came to Israel way back in '73, which was, you moved here in Aliyah. Was that because you got an offer, or because you were a Zionist, or why?
I guess, first of all, a lot of us who grew up in America, in the Jewish community, had this pull towards Zionism, to coming to Israel. And that was strengthened a lot when I was in medical school, and I took a trip in one summer to visit Israel.
Which year was that?
This was in 1965.
I see. Before the Six Day War.
Way before the Six Day War. I was a medical student, and I came here and I just completely fell in love with the country. And then I went back to my studies, and about a year or so later I met the person who would ultimately end up being my wife, who actually had been in Israel at exactly the same time and had fell in love with Israel exactly in the same way. And we sort of, at the spur of the moment, decided that's it.
And she was American too.
American, bro. Yeah. She has Israeli roots because her mother actually was born in Israel.
I see. So that's why Sippy.
Sippy,
yeah.
right.
So you had that idea in mind when you're finishing your MD and PhD.
Right. And then the Army came along, American Army came along, and they were still fighting the Vietnam War. And anybody who had gone through medical school had essentially gotten a deferment from the Army. So people in my class, for instance, who finished all had to go and got drafted into the Army when they finished.
But you served in the labs, in the military labs.
So I was lucky, and I got accepted to what was known as the Public Health Service, which is a branch of the military that allows you to do your service for 2 years, but not armed services. And I was fortunate to work at the National Institutes of Health in Washington, and that way fulfill my service to the United States. And then right after that, immediately after that, in fact, the Army actually shipped us to Israel.
Really?
Yeah.
But had you decided to be a researcher then, or because you came immediately to Hebrew University as a researcher?
And when I went to, my background is that I first studied math at MIT.
Oh, at MIT, that's right. Then you went to NYU.
And then I decided that I wanted to be a doctor. And as soon as I got to MIT, they offered this program to MD PhD program.
Oh, that's just when they were starting it.
Right. This was the first time that the program was offered. And I was the first person in America to finish with a doctorate in that program.
I didn't know that. Huh?
Yeah. And so already in medical school, I had a lot of research experience, and I realized then that this was my calling rather than the clinical work. Yeah, actually, I think that the ultimate reason I didn't go into clinical work was because I found I didn't really have the proper empathy towards patients.
I have a different explanation for mine. It wasn't so much the lack of empathy, but when I dealt with, I was in medical school. When my brother and I were in medical school, I didn't have the diagnostic skills.
Right?
That's what I discovered. Too abstract. Philosophy is fine.
I can understand that.
So it depends on what your disposition. You think because you're smart you should be in medicine, but in fact you're not suited to it.
Yeah, I discovered that all my patients lost their patience with me. It was not for me.
Yeah, no, it's interesting how we come to those different discoveries. I have the joke that I saved people's lives by leaving medicine. But getting on to your research, did you get right into genetics very quickly? Or?
So there also I was very fortunate. I went to a laboratory at the NIH where the laboratory of Gary Felsenfeld, and he was working on DNA. And really there weren't that many laboratories doing this so-called new molecular biology. And not only that, but he was doing this molecular biology on DNA of animal cells, of human cells. And up until then, most of the work had been done in bacteria. So it was a real opening, a real opportunity, which maybe I didn't realize it then, but looking back at it, it was a real golden opportunity.
And so you then took that, and so you must have been one of the pioneers in Israel then when you came here.
So when I came to Israel.
When did you get the Brenner chair? That comes later.
That was much later, right?
In the late 80s?
Sometime in the 80s, I don't remember when. When I came to Israel, I don't think there was anybody working on the DNA in animal cells, and there was certainly nobody working on what we called then chromatin, which is the structure of, it's the DNA together with its packaging, with its structure. And we were one of the first laboratories in the world to work on this, not only in Israel.
So how did you get into this? I mean, you make your mark as one of the famous scientists in the world, not just in Israel. You have the Wolf Prize, you got the Israel Prize earlier. You're very esteemed, but you make it in a particular area of the transformation of messages, etc. How did you get into that specific area?
So again, when I was at the National Institutes of Health, we were working on something called chromatin, this structure.
Right.
And we came to the conclusion, which was really a big breakthrough in understanding, that if you try to figure out what controls genes in an animal cell, the most important thing is how the genes are structured. And we sort of discovered that all of the DNA in an animal cell is packaged so that it's really not accessible. So all the machinery of the cell that's needed to read the genes, to read the information, can't get to the DNA.
So it's a closed system.
It's closed. And when a gene is turned on or used, there's a process of opening it up. And so this important idea of accessibility we developed in these studies. Right?
I see.
And this is true until today. I mean, the concept is one of the basic concepts of modern molecular biology, that gene regulation in animal cells at least, and in many other organisms, is controlled by what we call accessibility. And so with this, we came to Israel. We came about a month before the Yom Kippur War, which was not only a big shock, but made everything a little bit more difficult. It was very, very difficult to get the lab started.
I was here at the same time.
You were here also, so you know what it was like.
Well, no, I wasn't. I was just here for 6 weeks, so and then I left. So I wasn't here during the war.
Yeah. So it was hard to get things started, but And we started working in that direction. And that ultimately led us to this idea about DNA methylation, which then occupied our laboratory for the rest of our lives.
And that's what, well, you're called the father of DNA methylation, of course, in all the literature when you read about it. Can you briefly explain DNA methylation, and then we'll take a break?
Okay. I guess the simplest explanation of DNA methylation is that you need in the body, because we have multicellular body, every cell is doing something different. But every cell has the exact same genetic information, the same book, the same encyclopedia that we got from our parents. And there have to be mechanisms for controlling these genes, which ones are on, which ones are off in every particular cell. What's on in an eye cell is not on in a skin cell, and what's on in a liver cell is not on in a brain cell. And there has to be mechanisms for controlling this. And basically DNA methylation is the father of these mechanisms. It's a chemical switch.
Turns it on.
Put it on to a piece of information, it turns that information off. You take it off that information, it turns that information on. So it's a real switch.
We'll take a break now and come back to switching.
Okay, great.